Chapter 12, The Cell Cycle. This chapter will be on asexual reproduction, mitosis. In this chapter, the life cycle of a cell culminates in division, and we will learn the 4 phases of a cell cycle. We will also talk about how cell division takes place via mitosis, and then a new word, cytokinesis, is the separation of daughter cells. And then we'll talk about control of the cell cycle, and lastly, what happens when cells do not obey the cell cycle. In other words, what happens when we have out of control cell division, which is referred to as cancer. So, the introduction to the cell cycle. So when we talk about cell division, what are we talking about? We're talking about the formation of new cells, and these are formed by splitting of pre-existing cells. Now, we can have cell division in several ways. We can have it with fertilized eggs. We have embryos and we have sperm and egg, which are gametes, and they unite, forming a fertilized egg, and this is called meiosis. We'll talk about that in the next chapter, but for this chapter, we're going to talk about the cell division of somatic cells. Somatic cells are body cells, like eyeball cells, cheek cells, stomach cells, skin cells. All of those divide asexually, mitosis. After mitosis, the daughter cells separate. In other words, the cytoplasm in each cell becomes distinct, and this is called cytokinesis. If we look at the word cytokinesis, cyto, the prefix means cell, and kinesis means movement. Cells moving apart, cytokinesis. And at the end of mitosis, we have 2 daughter cells, and these 2 daughter cells are genetically identical. Mitosis, if one were to describe it, it is the genetic material is copied and divided equally between 2 cells. So in this cell cycle, a chromosome is replicated, and then during mitosis, this material is separated equally. So what is the purpose of mitosis? Is to separate replicated material. So that if someone were to tell me what mitosis is, that's what I would tell them. It's the separation of sister chromatids. In mitosis, each daughter cell is genetically identical to each other and to the parent. Whereas in meiosis, when we have sexual reproduction, every single cell is genetically different, and that's really important. It almost reminds me of a test question. So how do cells replicate? Well, there's 3 basic steps to replication, and keep in mind that when we talk about replication, it's fundamental to life itself. So the first step is copying DNA, which happens during the cell cycle, separating the copies, which happens during mitosis, and after mitosis, we have dividing of cytoplasm, which is called cytokinesis. So let's start off with what is a chromosome. Chromosome literally means colored body. Chromo, colored, some, body. And it's single long double strands of DNA. And these linear strands of DNA are typically wrapped around proteins called histones. And then on the DNA, we have a lot of base pairs, like all the nucleotides lined up. So on a chromosome, we can also say that there are many genes. So what is a gene? A gene is a region of DNA in a chromosome, and it's going to code for specific RNA that in turn will code for a specific protein. Because usually when we talk about a trait, we're talking about a type of protein. Before mitosis, all chromosomes are replicated, and they're replicated during the interphase part of the cell cycle. So right before mitosis, chromosomes condense and they become very compact structures. In fact, they look like the iconic X that you see on biology posters. And one copy of the chromosome will go to one side and the other one will go to the other. And this is mitosis. Each double-stranded DNA copy is called a chromatid. We call them sister chromatids. And chromatids are attached along the entire length by proteins called cohesions. But when we see the iconic X, where they cross, where one replicated strand is crossing the other replicate of itself, is at the centromere. See, these are sister chromatids. Here's one, and here's a replicated one, sister chromatids. When they're attached at the centromere in the middle, they're called sister chromatids. The minute they begin to separate during mitosis, then they're called chromosomes. In figure 12.1, you can see an unreplicated chromosome, a replicated chromosome, and then lastly, a condensed replicated chromosome. So notice right before mitosis, the chromosomes condense into that iconic X. But before that, they're very stringy and fine. They almost look like that spray that you spray out of the cans, those streamers. Cells alternate between M phase and interphase. Doesn't M phase represent mitosis, and later on it'll represent meiosis? But then we have interphase. Well, what is interphase? Interphase is the non-dividing phase, and this is where chromosomes are uncoiled and where they grow and they prepare to divide. This is also the time that chromosomes actually replicate their chromosomes. So cells spend most of their time in interphase. Chromosome replication occurs only during interphase. So S phase is when DNA replicates. This reminds me of a little story of my brother. When he was really young, like during the summertime here in El Paso when it's so hot, over 100 degrees, he would pretend to be Clark Kent, and so he would wear a jacket and underneath he had a towel. And that towel was his cape so that he turned into Superman. And he asked my mom to put an S on his cape. And one time I was so angry with him, I said the S stands for stupid, not Superman. But the truth is, what does S stand for? Synthesis, DNA synthesis. So synthesis is just a fancy word for make. So this is where DNA is replicated. So the cell cycle, what can we say about it? It's very orderly. It occurs from the formation of all eukaryotic cells, like cells in our own body. And during the cell cycle, this is when our chromosomes replicate, and it gets the cell ready for division. Now, on both sides of the S phase, we have gap 1 and gap 2. So there's two gaps, G1 and G2. G1 comes before the S phase and G2 comes after the S phase. If we were to look at this figure and we were to say that the cell would go through cell division within 24 hours through the whole cell cycle, you would see that most of the time the cell is in interphase, gap 1, S, and gap 2. The cell cycle in total represents 4 phases. What are the 4 phases? M phase is 1, G1, S, and G2. It is true that of the 4, 3 of these are interphase. G1, S, and G2 are all part of interphase. The G1 phase decides to begin replication and transitions into the S phase. And the G2 phase is the time that the cell uses to prepare for mitosis. Figure 12.3 shows the cell cycle. I particularly love this figure, and you're going to see it several times, and that indicates to you that this is a very important figure. So the cells, normal cells, must obey the cell cycle. Notice that interphase is composed of G1, S, and G2. and this composes the greatest length of the cell cycle. The smallest part is mitosis, the asexual reproduction of one cell into two daughter cells. To watch this video, go back to Blackboard in your presentation so that you can see the full video. So what happens during mitosis? It says mitosis consists of two distinct events. It consists of mitosis, the division of replicated chromosomes, but it also consists of the division of cytoplasm so that we have two distinct daughter cells. So these would be our two distinct daughter cells. So if we look at an overview that's in your book, we see in figure 12.4 an overview. So we see interphase where chromosomes are replicated, and you guys know by now that chromosomes are replicated during the S phase. So we start with 4 unreplicated chromosomes, and they just look like single strands. They look like pipe cleaners, blue pipe cleaners. And then we see replicated chromosomes. In other words, we go from a single to a replicated chromosome, but this still counts as one. So then we notice that they condense in the third figure of the cell so that they're very visible with the naked eye as the iconic X. And then during mitosis, they separate into two identical daughter cells, each with the same number of chromosomes that they began with. So if you have a cell that starts off with 10, then the daughter cells will have 10 chromosomes. If the parent cell, that one parent cell, has 50 chromosomes, then the two daughter cells will end up having 50 chromosomes. If you start off with 20 chromosomes, yeah, you're right, the two daughter cells will have 20 chromosomes each. So you end up with the same number of chromosomes that you begin with. What happens during the M phase? Well, when DNA starts wrapping around the proteins called histones, then it condenses to the iconic X that we usually see. And because of this, we call it chromatin. Chromatin refers to both DNA and proteins combined, or a DNA histone complex. Usually during interphase, the chromatin is relaxed. It looks like very thin spaghetti, but it condenses as the cell gets ready for mitosis. So during the S phase, the chromosomes are replicated and hopefully no mistakes are made. Mitosis begins when the chromatin condenses, and we have two sister chromatids. And what is the purpose of mitosis? Is to evenly divide the sister chromatins, or the genetic material, so that we now have two genetically identical daughter cells. Now, I don't include pro-metaphase for those of you who read your books very carefully. I just refer to the phases of mitosis as pay me anytime. My mnemonic device is pay me anytime. Prophase, metaphase, anaphase, and telophase. So when if I ask you any questions, I will never ask for an answer that includes the phase pro-metaphase. Now, even though we see them as separate phases, notice that the slide mentions that they're continuous. So notice here we have a cell, and then the cell, all of that genetic material condenses. We have replicated chromosomes, and so now we're starting mitosis. So the first phase is prophase. In prophase and prometaphase, notice that the spindle fiber apparatus is forming and centrioles are migrating to opposite poles. Also notice that the nuclear membrane is beginning to disintegrate and falling apart. And it must, because if they're contained within the nuclear membrane, then how will the chromosomes be able to migrate? So then by metaphase, all of the chromosomes line up in the middle. So notice that there's a lot of spindles coming out from the centrioles or centrosomes that have migrated to opposite poles. But the ones that are connected to the chromosomes are called kinetochore fibers or microtubules, and they're the ones that will help separate the sister chromatids and guide them to opposite poles. Notice in the next phase, anaphase, sister chromatids actually separate. And in this part here, when they start to separate, that means that now, instead of being called sister chromatids, they are now chromosomes. And then by telophase, they're actually forming nuclear membranes around each of the chromosomes that have migrated to opposite poles. And then they start to pinch off in between the two nuclear membranes, and they pinch off to form two daughter cells. And each of these daughter cells will be genetically identical. Okay, so let's go over the phases again. Prophase, pay me anytime. Pay me anytime. Prophase, metaphase, anaphase, and telophase, and then it ends off with cytokinesis. So in prophase, chromosomes condense, and the spindle apparatus begins to form. So the spindle apparatus consists of microtubules, and this is also referred to as the microtubule organizing center, or MTOC. So when you read about MTOC, you know that you're talking about spindle fibers, or you're talking about microtubules. They mean the same, and the words or terms are used interchangeably. So there's a plus n side to the microtubule. And notice here our first type of microtubule are polar microtubules. And these extend from each spindle pole, and they overlap each other. So then we have the centrosome, and in the drawings the centrosome looks like that bright sunny region that you see migrating to each end of the cell or the poles. Okay, during prometaphase, or what I call prophase, the nuclear envelope continues to break down and chromosomes attach, but now they attach to a second type of microtubule. So we know about astromicrotubules. Now the second type are kinetochore microtubules. And these start aligning themselves, pushing and pulling, but the chromosomes eventually meet in the middle. And these kinetochore microtubules are attached to the chromosomes. During metaphase, all of the chromosomes are lined up in the middle of the cell. Middle. That's how I always remember it. They talk about a metaphase plate. That would be like the equator of the cell. Each chromosome is held by a kinetochore microtubule, and this is what will be used to guide the chromatids that are actually being separated and traveling to other ends. The astromicrotubules are holding the poles in place. During anaphase, actually the sister chromatids split, and each of the sister chromatids are pulled by separate microtubules, or these spindle fibers, towards opposite poles. And this is what's going to create two identical sets of daughter chromosomes. So two forces are pulling the chromosomes apart. The kinetochore microtubules shrink, and then a motor protein is actually pushing the two poles away from each other. In telophase, the nuclear envelope begins to form again, and chromosomes begin to decondense. Instead of looking nice and thick and visible, they're beginning to look like thin spaghetti all over again. Mitosis is complete when two independent nuclei have formed. So the last stage is for the cytoplasm to separate, and this is called cytokinesis. I really appreciate Table 12.1 There's so many new terms that we're learning for cell division, and this table has a long list of actually structures and their definition. Please make sure that you look over these terms and you understand them. This is another short video, so go back to Blackboard and refer to the presentation that I have there so that you can view it. How do chromosomes move during anaphase? Well, they start to walk along actually the kinetochore microtubules. So believe it or not, the kinetochore microtubules are not moving at all. They're laying straight. In fact, maybe the best analogy is to say they're like railroad tracks. So they remain straight and they're not moving or anything during actually the part during anaphase where we see chromosomes moving towards opposite poles. So how is it that the microtubules become shorter and shorter over time? They shorten because tubulin subunits that compose the microtubules are lost from the plus ends. In other words, the railroad track begins to disintegrate as the chromosomes walk along the tracks. Proteins from the kinetochore attach to the ring that surrounds the kinetochore microtubule, and as it walks along, the plus end disassembles and the ring moves along the microtubule. How is it that we know this? We know this from a very simple but elegant experiment. So here we see that the chromosomes and actually the spindle fiber apparatus is fluorescing, one in blue and the other in yellow. And then notice on the right hand side, a section of the spindle fibers has been bleached. Now, if the spindle fibers are actually moving during anaphase, then we would expect that bleached region to move towards the pole. But in fact, in the last frame, you see that it remains stationary. It is not moving at all. So what is happening as the chromosomes walk along the track or the microtubule, the plus end of the fiber begins to disintegrate. Kinetochores are linked to retreating microtubule ends. The complex of proteins attached to the centromere region actually will walk along the fibers, and these fibers extend from the kinetochore, and they're tethered to the ring surrounding the kinetochore microtubule. As this ring moves across, then the plus end of the kinetochore microtubule begins to fray or disassemble. And it looks like actually the fiber is becoming shorter and shorter, which in fact it is. I love figure 12.7 because you can see the ring of the kinetochore and how it is walking along the microtubule fiber. Notice that as the ring moves and walks along carrying the chromosome, that the plus end or the tubulin subunits begin to disassemble. And this is why the fiber becomes shorter and shorter. Cytokinesis results in two daughter cells. Cytoplasmic contents increase in number and volume. During cytokinesis, cytoplasm divides to form two daughter cells. Now, cytokinesis is different if we take a look at plants versus cytokinesis that happens during in animal cells. So cytokinesis in plants, we have a formation of a straight cell plate. But if we talk about the separation of cytoplasm in animal cells, we see that it's pinched off like dough. It pinches off and the two daughter cells separate. This pinching is the result of a ring of actin and myosin, which actually contracts, and it becomes tighter and tighter, forming a cleavage furrow until the ring shrinks and tightens and cell division is complete. Figure 12.8 visually shows the differences between cytokinesis in plant cells versus animal cells. Notice here we have a cell plate forming in plant cells, but we have a cleavage furrow forming in animal cells. So what happens in bacteria? Bacteria doesn't really quite have mitosis. They use the term binary fission, bi meaning two. It's a process very similar to the mitosis that happens in eukaryotic cells. Remember that bacteria have a single circular chromosome, and this circular chromosome is replicated, and then it too migrates to opposite poles. But instead of microtubules, it has these protein filaments, and these protein filaments pull the replicated chromosomes apart. In this diagram here, you can see that a circular chromosome that is condensed is replicated, and this protein filament is pulling the replicated chromosome apart to opposite poles. Then you see a pinching or a constriction in the middle, that fission, and once it's complete, you have two bacterial cells. Let's now talk about control of the cell cycle. The cell cycle length can vary greatly among cell types, and I think intuitively that makes sense, right? That a cheek cell is very different from your brain cell or a liver cell, so we wouldn't expect them to really behave the same going through the cell cycle. I remember when I was little, my parents used to tell me, please don't drink any alcohol because your brain cells never divide. They never divide, and you can't afford to lose not one of your brain cells. So please don't drink any alcohol. And I used to say, okay, but we know differently now that the brain is very dynamic. That if cells do not divide, then we call that arrested state the G0 or the G0 state. Division rate can also vary in response to changing conditions, and variations in cell cycle length suggest that the cell cycle itself is regulated. Okay, so how did they find out that there were molecules that were actually regulating the cell cycle? So in the 1970s, researchers fused cells in different stages of the cell cycle, and this formed a single cell with two nuclei. And one nuclei often changed the phase of the other nucleus. And this is how they knew that there must be molecules regulating actually mitosis. So the hypothesis of this experiment is that cytoplasm of M phase cells have regulatory molecules that induce interphase cells to enter the M phase. In other words, they could get cells to mitotically divide. And what they did, if you look in your book, and it says here oocytes, is what they did is they took frog eggs and they got non-dividing frog eggs to actually divide. How did they do that? They injected the cytoplasm of a frog egg that was dividing, and they took that material and they injected it into another frog egg, and lo and behold, that cell also began to divide. So then they concluded there must be some molecule in the cytoplasm that is regulating the cell to divide. So we have this M phase promotion factors, and it's called MPF, mitosis promoting factors. So what they concluded was cytoplasm of M phase cells contain a substance that drives immature oocytes into M phase to complete maturation. In other words, there are molecules in the cytoplasm regulating the onset of mitosis. Okay, so what are the molecules that compose MPF, mitosis promoting factor, or M promoting factor? and it turns out to be cyclins. And cyclins are a family of proteins. So there's cyclins and cyclin-dependent proteins. So frogs are stimulated to divide by these proteins that cycle within the cell. Okay, so keep in mind that MPF represents M-phase promoting factor or mitosis promoting factor. So it says MPF is made up of two distinct polypeptide subunits. And remember, polypeptide is just another name for a protein. So what are the two units? It's a protein kinase and cyclin. We can say that this protein kinase is protein dependent kinase or cyclin dependent kinase. So it's protein kinase is CDK, and then we have cyclin. Okay, so this is how they did the experiment, and this is how they got cells to divide. What proteins are responsible for MPF activity? Well, we know that it's cyclin, and the kinase protein is cyclin-dependent kinase, CDK and cyclin. And these two subworkings together make up MPF, the mitosis promoting factor. Now, notice here, it says here that cyclin increases during interphase and it peaks in M phase before decreasing again. So cyclin cycles, the concentration of cyclin cycles during the cell cycle. This figure 12.11 is critical to understanding how these two units work together to control mitosis, MPF, the mitosis promoting factor. So notice that you see that there's two proteins, cyclin and cyclin-dependent kinase. Now let's look at the green part of the second subunit, CDK. Do you see that there's one phosphorus unit there? Well, it looks like there's a secondary site at the top for another phosphorus. And there are two, but when cyclin concentrations become so high, cyclin then binds to cyclin-dependent kinase. And what happens is it kicks out the top phosphate group. And when it kicks it up, it's like a light switch, it's been turned on. And it has this domino effect. Remember, energy is transferred in the cell in two ways, the movement of electrons and the movement of phosphate. And in this case, the movement of phosphate starts a chain reaction of phosphorylation that turns on mitosis. So if we look at the graph below, we see that the x-axis is in fact the cell cycle, G1S, G2 M phase, G1S, G2 M phase, and so on and so forth. The y-axis is MPF component concentration. So notice that cyclin increases during interphase and peaks during the onset of mitosis, then rapidly degrades during mitosis, and then again it starts to increase during interphase. And that is why cyclin cycles, the concentration cycles during the cell cycle. When concentrations are high, notice that it binds to CDK, the green line at the top of the chart. And when it binds with CDK, then cyclin-dependent kinase catalyzes phosphorylation of other proteins. In other words, that top phosphate group is released, and now we have a cascade of events leading to mitosis. So how is MPF turned on? Well, it has two phosphorylation sites, as you could see in the previous figure. And when cyclin becomes very high, then it binds to CDK. And this, it says both, so prior to that, the two phosphates in cyclin inhibit the kinase. that when one phosphate group is removed, it activates the protein, and this leads to chromosome condensation and formation of mitotic spindle fiber apparatus. Okay, so how do we turn it off? We know how it's turned on. Cyclin binds to CDK, and because of that, one of the phosphate groups is released. Now, how do we turn it off? Well, there's negative feedback occurs when a process is slowed or shut down by its product. So in this case, we need to destroy the enzyme that is a common way to control the cell process. So we need to destroy cyclin. So notice during mitosis, an enzyme complex is activated and it attaches to cyclin during anaphase, and during anaphase cyclin then becomes destroyed and deactivated. Cell cycle checkpoints can arrest the cell cycle. Here, when we say arrest, we mean to stop. It can stop the cell cycle. So there are 4 cell cycle checkpoints, and these cell cycle checkpoints are really important. The best analogy I can give is national security. Isn't it true that we have people that regulate the crossing of people at our bridges? For example, here in El Paso, we have people along the El Paso Juarez border. And then we also have another group of regulators at the airports, right? And they're regulating the flow of people coming in and out of the city via plane. And then if you go 100 miles or 200 miles away from the border, you have another checkpoint. Here we have checkpoints in New Mexico, and we also have them in East Texas as you move east. So we have other checkpoints if you're traveling by car. So for national security, we have multiple checkpoints. So it makes sense. The same idea would be used at the cellular level. There are multiple checkpoints. And these regulatory molecules decide whether a cell can proceed to the next step. Now, cells that divide without any control can oftentimes become a tumor. And so this is what leads to cancerous cells. Figure 12.12 shows the 4 checkpoints. Notice the first checkpoint is in G1. G1 is right before DNA synthesis. The second checkpoint is right before mitosis at the end of G2. And then the last 2 checkpoints are during mitosis itself. These checkpoints make sure that chromosomes have attached to the spindle fiber apparatus and that chromosomes have actually divided equally. So let's go over the first checkpoint real quickly. So there are 4 factors to look at the G1 checkpoint. Out of all the checkpoints, G1 is first and foremost the most important checkpoint. This could be an important test question. So what are the 4 factors that are being checked at the G1 checkpoint? Size, availability of nutrients, social signals, and damaged or undamaged DNA. If DNA is physically damaged, then we have a p53 protein, and this activates proteins that will actually stop or arrest the cell cycle. It could almost even program the cell for death, and this is called apoptosis. So p53 is considered to be a protein that is a tumor suppressor. It stops tumors from growing. It stops damaged cells from continuing in the cell cycle. Now, if there is damage to p53, then this can lead to uncontrolled cell division. The second checkpoint occurs after DNA is replicated during the S phase, and this is in the G2 phase. So if there's any mistake or damage during DNA replication, the cell will be arrested again. So NPF will not be activated and the cell will remain in the G2 phase. The final two checkpoints are during mitosis. One is between metaphase and anaphase, and then the last one is between anaphase and telophase. One is to make sure that spindle fibers, the kinetochore microtubules, have attached properly to the replicated chromosomes, and in anaphase and telophase is to make sure that genetic material has separated equally. Again, we see the 4 checkpoints. So please make sure that you take a look and you're very comfortable knowing the 4 checkpoints of the cell cycle. Okay, so the last part of this chapter is what is cancer? Cancer is out of control cell division. Cancer is when none of the checkpoints are obeyed, or when damaged cells are not caught and they continue moving within the cell cycle. 40% of all Americans will develop cancer, and cancer is a complex family of diseases, and it can attack all different kinds of tissues within the body. There's over 200 types of cancers, and they arise when these checkpoints have failed. So if we take a look at this graph, we see blue and red bars. Blue represents estimated new cases, and red bars indicate estimated deaths. So I think the good news by looking at this graph, it shows that just because you have cancer does not mean that you are programmed for death, that there are a lot of people that are surviving cancers. Now, there is one cancer that does not have a good rate of survival, and that is pancreatic cancer at the bottom. But for most cancers, almost including lung cancer, you see that if there's proper treatment, there is hope for survival. Cancerous cells have 2 types of defects related to division. Defects activate proteins required for cell growth when they should not be active, and they prevent tumor suppressor genes like p53 from shutting down the cell cycle. So these are the 2 ways that we can have damaged cells. Now there's 2 types of tumors. There's malignant tumors and benign. Malignant, or mal, must have a Latin origin, mal meaning bad. These are cancerous and very invasive cells. These are tumors that can spread throughout the body via the blood and lymph system, and this is called metastasis when they're not anchored and they move around in our body. The other type of tumor is a benign tumor. It is non-cancerous and non-invasive. Figure 12.14 shows the difference between benign and malignant tumors. Benign tumors are at the top. Notice you see rapidly growing cells, but they have not entered into actually the circulatory system indicated by red channels, nor have they entered into the lymph system indicated by green channels. But if you look at the malignant tumor, notice that you have a lot of dividing cells, and they enter into both the circulatory system and into the lymph system, and they're metastasizing and invading other regions of the body. The last part talks about cancer involving loss of cell cycle control. So I guess what I'd like to mention is we've been talking about mitosis, or asexual reproduction of a single cell. That if you think about it, let's say when you scratch yourself or cut your skin, it's not just one cell dividing, but many cells. So how can we get a lot of cells to divide at the same time? So they're under these social control signals and growth factors. This will not be on the test, but you may have this in upper division classes. So this is your first introduction. So there are these messengers within the cells to get many cells to mitotically reproduce at the same time. So in addition to these growth factors are other proteins that can be found within the cell, like E2F protein and the RB protein. And these proteins work together along with cyclin and cyclin-dependent kinase to actually get cells to mitotically divide. And here in this diagram, for those of you who are interested in actually these social signals, you can see that there are many signals in actuality that must be passed for the cells to mitotically divide. So, how do social controls and cell cycle checkpoints fail? Cells can become cancerous when social controls fail. Cells begin dividing in the absence of a go-ahead signal. In some cancers, G1 cyclin is overproduced. So we can have social control failure, cyclin is overproduced, RB is defective. So there's a lot of ways in which we can get cells to reproduce over and over again. And this is the end of this chapter.